Imagine a human population arriving on a high-radiation planet. The biological substrate breaks down, and unprepared cells rapidly mutate and rupture. Darwinian evolution is unequipped for this timeframe. It relies on the macroscopic environment to act as a slow, stochastic filter for microscopic genetic variations. This bottom-up flow of biological data creates informational friction. There is an immense thermodynamic distance between your DNA and the lethal environment. Because of this, the biological clock becomes excruciatingly slow, bogged down by random noise that destroys most upward-moving genetic signals. In a highly lethal environment, the probability of population extinction approaches unity long before protective alleles can successfully fixate into the genome. This thermodynamic slowness in informational flow ensures that natural adaptation cannot keep pace with the hazards of deep space. Complex organisms are mathematically required to maintain low mutation fidelity to prevent widespread genomic instability. If a species attempts to speed up its adaptation by increasing its mutation rate, it inevitably hits a mathematical wall known as Eigen's paradox. Crossing this limit triggers an immediate error catastrophe. Deleterious mutations outpace the capacity of natural selection to purge them, causing the rapid degradation of the genetic code. This is compounded by Haldane's cost of selection. A natural genetic sweep requires a specific number of selective DATs, and in extreme environments, that required death toll easily exceeds human reproductive capacity. Surviving on Mars or the Jovian moons demands multigenic adaptations that natural biology cannot provide quickly enough without triggering immediate population collapse. Escaping this thermodynamic stalemate requires humanity to bypass environmental pressure entirely, using conscious intent to initiate a synthetic phase transition. To solve the informational friction problem, we have to look at the physics of how information density behaves within biological systems. This is modeled through the infinite fractal descent framework. Its core fractal density equation balances the rate of descent, gamma, against the action-tension constant, sigma. When the macroscopic interaction scale expands, the informational density output shrinks to a fraction of a percent. This low density explains the slowness of natural evolution. The signal of survival is diluted across the vast fractal distances between the ecosystem and the double helix. The synthetic phase transition solves this. By utilizing directed computational power and tools like CRISPR-Cas9, we manually collapse that interaction scale. We bypass the intermediate ecological and organismal layers entirely, injecting localized informational intent directly into the atomic geometry of the genome. This targeted scale collapse turns adaptation into a high-speed, computable phase transition. This chart shows the evolutionary acceleration factor, modeled as a function of the fractal interaction scale. Tracking the logarithmic interaction scale on the x-axis reveals a massive density spike as the descent reaches the genomic base pair level. This resulting compression gives us an exponential evolutionary acceleration factor, allowing geological epochs of adaptation to be condensed into single gestational lab cycles. The primary physical challenge is ensuring that a microscopic atomic edit does not cause unforeseen side effects when scaling back up to the whole organism. This diagram illustrates infinite fractal descent geometry. We use a computational technique called renormalization to rescale the edit and preserve its functional geometry. Morphometric flow moves precisely down the Fibonacci spiral, ensuring macroscopic intent maps onto the microscopic target without functional degradation. Precise renormalization ensures that the biological system transitions cleanly into its new steady state, shielding the edit from the stochastic noise inherent in natural systems. This math brings us to the concept of chassis terraforming. It is thermodynamically more efficient to modify the human biological vehicle than to attempt to re-engineer an entire planet. Take the extreme biological stressors of the Martian environment, specifically ionizing radiation and microgravity. In an Earth baseline human, high radiation induces DNA strand breaks, while microgravity inhibits osteoblast differentiation, destroying bone density. The synthetic solution for radiation is the targeted integration of the D subgene, originally observed in tardigrades. D sub acts as an intrinsically disordered protein. It physically coats the nucleosomes to provide electrostatic shielding against the hydroxyl radicals that cause DNA damage. In the IFD framework, this edit is a deliberate increase in the genome's action-tension constant, designed to preserve informational density in harsh environments. By engineering the chassis to negate environmental stressors, we render the hostile environment thermodynamically irrelevant. Planetary surface conditions are only one application. The long-duration transit through deep space requires a fundamental departure from baseline human metabolism. This bar graph shows the causal budget allocation, mapping the shifting internal resource economy across different human variants. The deep space variant heavily alters its causal budget, shifting the dissipative balance away from maintenance and toward translation. To achieve this efficiency, engineers utilize the synthetic manipulation of uncoupling protein 1, or UCP1. UCP1 uncouples fuel combustion from ATP production, lowering metabolic demand. This is paired with the deletion of vestigial organs and the recoding of metabolic networks to recycle urea and CO2 directly back into biosynthetic pathways. This combination of metabolic uncoupling and internal waste recycling effectively creates a closed-loop system, reducing the resource cost of maintaining biological coherence in the void. Looking at the entire arc of this evolutionary phase transition, we see a fundamental shift in system entropy. Natural selection requires an immense thermodynamic cost, driven by mass population death. Synthetic evolution operates with calculated precision. We harness negentropy, derived from high-density computational simulations. This allows the human biological algorithm to jump directly to optimized fitness states. We can visualize the totality of biological potential as a dimensional projective manifold, mapping out possible genetic states. Applying synthetic intent curves this manifold, allowing the biological system to fall into its optimized multi-planetary configuration without crossing fatal evolutionary valleys. Surviving the extremes of the cosmos is no longer an insurmountable biological miracle. It is a predictable, geometric mathematical output. As our computational capacity scales toward infinite descent, the boundary between natural and synthetic biology dissolves. The biological clock is no longer bound to the slow rhythm of planetary ecosystems. Multi-planetary expansion has become a calculated computational process, unfolding according to the geometry of the biological manifold. Through the mastery of the synthetic phase transition, humanity has ensured its consciousness will continue to evolve, adapt, and resonate across the stars.